there aren't any questions to start with, we'll go to screen sharing, and we're going to start by looking at the bacteria page that we looked at the other day and continue with where we were. Oops, I skipped a little ahead. So when we left off, we were talking about the idea that bacteria don't reproduce by sexual processes. And if we look at how the cells reproduce, they don't reproduce by mitosis or meiosis. Those two processes only occur in eukaryotic cells. So let's look at how a bacterial cell can reproduce. And let's keep in mind what I said the other day. This whole process can happen very rapidly, in about 20 minutes or 25 minutes if conditions are optimal for the bacteria cell. Now, conditions aren't always optimal, of course, so a lot of times they reproduce more slowly, but if everything is perfect, bacteria cells can reproduce 20 minutes after they were produced. So a very rapid rate of reproduction. And really there's a couple of steps. If we think about down here at the bottom, this is the size of one of these bacteria cells. So when the cell is getting ready to divide by binary fission, the cell grows, and then as it grows, it gets longer. And the cell replicates its DNA. Replication is fairly similar to what we see in eukaryotic cells. The details of where replication starts are a little bit different because remember, bacteria cells only have one chromosome. Eukaryotic cells typically have chromosomes that occur in pairs. But the basic process is very similar. And one way it's similar is if the DNA is correctly replicated, when it's done, this bacteria cell has two chromosomes that are genetically identical. Now, that's discounting any errors or any mutations that might have occurred, but if everything is right, we have a glob of chromosome here on the left, we have a glob here on the right, those are genetically identical. Now, what happens is the cell starts to pinch in from the outside. So remember when we talked about mitosis in animal cells back in 105, we talked about that cleavage furrow. So this is similar to that cleavage furrow. Notice there's actually two parts of the cell that are pinching in, the cell wall and the plasma membrane. And as they pinch in and as the cell continues to get a little bit longer, that pushes these two chromosomes apart from each other. So when the cell wall and plasma membrane start to pinch in right in the center of the cell, that starts to divide these two chromosomes into different cells. Eventually, the cell wall and the plasma membrane go all the way across, and when they go all the way across, we now have two distinct cells. Now, this shows the cell separating, but notice that cells don't always separate. Different bacteria cells have different patterns. So remember the other day we talked about the different shapes of bacteria cells. And one of the shapes we saw was a spherical cell called a coccus cell. There's one type of bacteria that when it reproduces, when it divides by binary fission, the cells stick together in clumps of two. So if we look at the two cells here, they would be drawn circular. This is notice if you think about it, this is a bacillus-shaped cell, but clumps of cells stick together. So this is a genus of bacteria called Diplococcus. So Diplo tells us that two cells stick together. Coccus tells us the shape of the cells. So sometimes when we look at the clumps of cells that stick together, that tells us something about how binary fission occurs. Now notice these two distinct cells that we have here at the end. These two cells are genetically identical. as I said, unless there's some error during replication. And so errors do occur with pretty regular, on a pretty regular occasions. So for instance, there's one bacteria that you've probably heard of called E. coli. Well, there are different strains of E. coli. You could maybe think of the different strains of E. coli as being like different breeds of dog. They're all the same species, but there are slight genetic differences because of these mutations. Some strains of E. coli cause things like food poisoning. Some strains don't. They're harmless. Now, when we look at bacteria, in bacteria, reproduction, which happens almost always by binary fission, there is one other process called budding, where one bacteria cell kind of grows out of the side of another bacteria cell and then pinches off. But usually reproduction is by binary fission. Reproduction is always separate from sexual processes that change the bacteria DNA. Bacterium is just plural. So if we're talking about one bacteria cell, technically it's a bacterium. Now, notice these sexual processes are sometimes referred to as horizontal gene transfer. So when you think of horizontal, think of sideways. Back in 105, we talked about genetics. We talked about, making Punnett squares. In this class, we've been talking a little bit about changes in the gene pool. Notice when we look at what happens from one generation to the next, this is sometimes called vertical gene transfer. So when we say vertical, that's up and down. So think of those pedigrees that we looked at, where different generations were at different lines on the pedigrees. Whereas horizontal gene transfer, all these sexual processes that we're going to talk about in bacteria, these are all processes that occur within a single generation. So bacteria can do something that typically eukaryotic cells can't do. Imagine that you have a genetic resistance to a disease. I can use my family as an example. Most winters, I end up getting the flu. Most winters, my wife doesn't get the flu. So I'm pretty sure she has some genetic resistance. There's something in her immune system that helps her resist getting the flu. Now, keep in mind, what fights off the flu or other illnesses in your body? Proteins. So we know that the instructions for making proteins are in the DNA. So this is a genetic characteristic. So if people could do horizontal gene transfer, that means my wife would somehow be able to transfer the instructions for making these proteins that give her a resistance to the flu. She could transfer that DNA to me so that I could become resistant to the flu. So bacteria can do that. So it's something that, really is very different than the genetics that we talked about back in 105. And what's even stranger is, if we think about genetics, if we work genetics problems, we're working genetics problems with people or with hens and roosters, or with bulls and cows, or with stallions and mares. We're working them with members of the same species. Some of these horizontal gene processes can transfer DNA, from one bacteria species to a completely different bacteria species. So this would be a transfer between a horse and a giraffe or something like that. And we don't really see that sort of process in eukaryotic cells. So we're going to look at three processes called transformation, transduction, and conjugation. There's another process called transfection. As far as we can tell, this doesn't really occur in nature. This occurs in lab experiments where we give a bacteria cell an electrical shock. And then giving it that shock allows it to pick up little pieces of DNA that are out in the environment. So let's think first about these little pieces of DNA. These are called plasmids, and we find them in many bacteria cells, not all bacteria cells, but many. And we can think about the scale of things here. If we look at a eukaryotic cell, if we look at a human cell, for instance, a human cell has about 23,000 genes. But an average cell in the human body only makes about 10,000 proteins. So in a person, what makes a red blood cell different from a white blood cell or an adipose cell or a bone cell? Well, what makes them different is which 10,000 genes do they use to make their proteins? Every cell in your body has the gene for a protein called hemoglobin, but only red blood cells in your body, only those cells use that gene to make the protein hemoglobin. In all the other cells, it's turned off. If we look at bacteria cells, now remember what I said, a typical eukaryotic cell ends up making about 10,000 different proteins, or maybe I should say uses 10,000 different genes. A typical bacteria cell only has about 10,000 genes. So really it uses everything it has. The smallest bacteria cells that we've discovered in terms of their genetic material have about 4,200 genes. As far as we know, there's no living organism with fewer genes than that. If we look at these plasmids, so this red in here represents that chromosome of the bacteria cell that's located in that nucleoid region. So that represents about 10,000 genes. The size of this plasmid is really exaggerated because this plasmid, if it's a really, really big plasmid, might have 100 genes, but it might have a lot fewer than 100. It might have 10 genes, might have 20. So the cell is 4,000 plus genes. This plasmid is less than 100. Now notice this little green hexagon here, we'll talk about that later, is a virus. And viruses, in terms of how many genes, are kind of like plasmids. Some viruses have just a few genes, some have 50 or 60. So we're going to look at transformation, transduction, and conjugation simply because, as far as we can tell, all three of these processes occur in nature, whereas transfection doesn't, only in a lab when we basically electrocute a bacteria cell. So let's look at conjugation first. And conjugation is a process that involves two bacteria cells. Now, one of the cells is called our donor cell. One of the cells is called the recipient cell. And the donor cell is going to transfer DNA to the recipient cell. Now, the donor cell has a plasmid called an F plasmid. And on this F plasmid, there are some genes, and the genes direct the building of a pilus. Remember, we said pilus are protein structures that stick out from the surface of the bacteria cell. And this pilus works kind of like a tunnel. And it's sometimes called a sex pilus because it connects two bacteria cells, and it's going to be used to transfer DNA. Remember, I said this is a sexual process. So what happens is the donor cell builds a pilus. This allows a few chemicals to flow back and forth between the donor and the recipient cell. And the donor uses several enzymes. And what these enzymes do is these enzymes untwist the DNA in this plasmid. Remember, DNA generally occurs in the form of a double helix. So this separates the two strands of the double helix. This does the job that topoisomerase and helicase and some of the enzymes that we talked about in 105 do during replication. Now, so instead of having double-stranded DNA, we have two single strands. One of the single strands of DNA is transferred into the recipient cell. One of the single strands of DNA stays in the donor cell. Now what happens in both cells? DNA polymerase comes in and builds the complementary strand. So we end up with a double-stranded DNA strand, a double-stranded DNA plasmid, back here. We end up with a double-stranded DNA plasmid over here. Since they were made from two complementary strands of DNA, again, if there are no errors, these two strands should be identical. So we've transferred some DNA from the donor cell to the recipient cell. At that point, the pilus separates, so the two cells are now different. Now notice one of the genes that gets transferred on this F plasmid is the gene for building a sex pilus and initiating the transfer of DNA. So now the cell that started out as a recipient, now this cell can be a donor and it can transfer DNA to other bacteria. So notice that plasmids are very important. We'll see another case where plasmids are involved in a moment, but notice that a lot of the genes for proteins that give bacteria antibiotic resistance are actually found in plasmids. They're not found in the chromosome. So one of the things this means is if you have some bacteria that are resistant to an antibiotic, they can transfer that resistance to other bacteria by transferring the plasmid. And not only can they transfer it to other bacteria of the same species, they can transfer it to other bacteria of completely different species. And this is actually one of the huge problems that we're having in medicine today. We have bacteria becoming resistant to many of the antibiotics that we normally use in hospitals. So a lot of research pharmacists spend their time trying to develop new types of antibiotics, either completely new drugs, or we have some antibiotics like penicillin, which actually comes from a mold, but we've developed synthetic versions of penicillin. And if you go back to 105, when we talked about chemistry and we talked about the different functional groups, one of the ways that we build new versions of penicillin is we take old functional groups off of the molecule and we replace them with a slightly different functional group. Now remember, on a molecular level, everything works by its three-dimensional shape. So what we try to do is build a version of penicillin that will still affect the bacteria the way we would like it to, but it's just different enough that the bacteria aren't resistant to it. Computers have made this job a lot easier, but it's still very tedious. Now let's look at a second process, which also involves transfer of DNA. Although this time, it's not a direct transfer from one bacteria cell to another. We have a bacteria cell that gets infected by a virus. And bacteria cells that are infected by virus, some of these viruses actually insert their DNA into the bacteria chromosome. So here we have our kind of peach-colored bacterial chromosome, and this black represents the DNA that came from the virus. Again, notice this is really exaggerating the size of the virus. The virus is maybe 50 genes long. The bacteria chromosome is at least 4,000, probably 10,000 genes long. Now, the prophage can go through a process called the lytic cycle. And in the lytic cycle, the prophage can break out of the chromosome, and it's hard to see. So let's increase the magnification. We'll go back down in a moment, but let's make this really big. So you can see there's a little bit of peach color inside this virus, that has now broken out of the chromosome. So this virus now contains a little bit of DNA from the original bacterial DNA. And when this virus reproduces inside the cell, all the new little viruses have that little sliver of the original bacterial DNA. So now what happens is once we have this bacterial DNA inside the viruses, after they reproduce, they're released from the bacteria cell. They usually rupture it and kill it. Notice how our diagram shows the plasma membrane is being destroyed in this area. That little pinkish purple line doesn't go down there. Now, when this virus infects a new cell, and this virus inserts its DNA into the new cell, the viral DNA is carrying that little sliver of DNA from the first cell that was infected. So this virus can transfer DNA from one bacteria cell to another. So we're still transferring DNA from one bacteria cell to another, but it's not a direct transfer. We have kind of a middleman, if you want to think of it, this virus. Now notice, viruses and plasmids have been used extensively in genetic engineering. If we can find a virus that will infect a cell, and we have a protein we'd like that cell to be able to make, we can put that gene for the protein in that virus, and we can try to insert it into the cell and get that cell to start making our protein. It's much easier to do with bacteria cells because in bacteria cells, almost all the DNA is used to make proteins. If we take a virus and we insert DNA into a human cell, the problem is in a typical human cell, only about 2% of the DNA is actually used to make proteins. So unless we have a way of getting that DNA precisely where we would like to put it, we don't necessarily accomplish anything. Our cell might pick up the DNA, but might never use that DNA to make a protein. So we're getting a little bit better at getting cells to make proteins that we insert the DNA for as we learn more about how genetics works. Now, finally, let's look at the last process, transformation. And transformation was first discovered back in the 1920s. And keep in mind, the structure of DNA wasn't discovered until the 1950s. And in the 1920s, a lot of biologists still thought that protein was the genetic material. Because, you know, if you look at DNA, all the DNA is the same. The only difference is whether it's got that A, C, G, or T. So there didn't seem to be enough variation in DNA to account for all the variation that we see in the phenotypes of living organisms. So in this case, a scientist named Griffith, no relation, was studying bacteria that can cause pneumonia. And you can't see one bacteria cell, but if you put them into some nutrient agar, if you put them on a petri dish and give them some food, and you let them grow for a few days so that they can divide, so that now instead of one bacteria cell, you have millions of bacteria cells, they produce a colony. And then you can see that colony. So Griffith took one particular type of bacteria that came in 2 strains. So remember what I said earlier, strains are slight genetic variations of the same species. And one of these strains produced rough colonies. When I say rough colonies, the colonies had kind of jagged edges when you looked at them. So he took some bacteria from the groups that produced the rough colonies. He put them into a hypodermic needle and he injected them into lab mice. And the mice were fine. He took some bacteria from the strain that produced smooth colonies. And the colonies had nice, even edges instead of being all rough and jagged. he puts some bacteria from these smooth colonies into a hypodermic needle, injected the lab mice, and they died of pneumonia. What Griffith was trying to do was find out exactly what was causing this difference between the two strains. And as I say, remember, at the time, he didn't know that DNA was the crucial material. So next, he took some bacteria from this smooth strain, the strain that was deadly, and he basically put them on a microscope slide and he heated them over a Bunsen burner. So they were dead. So the dead bacteria are shown in red. He put some of these dead bacteria into a hypodermic needle, injected them into some of his lab mice, and the mice were fine. Well, not too surprising. I mean, if the bacteria are dead, how can they cause pneumonia? Now, the last experiment he did, I'm not sure why he did it, but this was what led him to transformation. He took some bacteria from the smooth strain that caused pneumonia, and he heated them until they were dead. Then he mixed them with some bacteria from the rough strain. He injected them into the mice, and the mice got pneumonia and died. And not only that, when he did an autopsy of these dead mice, and they did autopsies of all of the mice that died, he didn't find these rough colonies. He found smooth colonies. But he was confused because the only bacteria from the smooth colonies had been dead when he put them in there. So he said that somehow these bacteria that made the rough colonies had been transformed. So that's why this process is called transformation. Years later, another group studied this process and realized that it was genetic nucleic acid, the genetic material, that was causing this transformation. Now notice, this means that bacteria can pick up DNA from their environment, because once he killed the smooth strain with heat, they were dead, and all that was left was some of the molecules, kind of like, you know, what you might find at the bottom of a cooking pan after you make dinner. So they just picked up DNA from non-living sources that were in the environment. And since then, we've discovered that bacteria sometimes shed these plasmids into the environment, and other bacteria cells can just wander by and pick them up. So all of these processes change the DNA of existing bacteria, but there are no new bacteria produced. So reproduction, you always have to make new organisms. Horizontal gene transfer, you just change the DNA of organisms that are already there. So that's what we have with transformation and transduction and conjugation. So notice in bacteria, sex and reproduction are two completely different things. Now let's think about where we find bacteria in the environment. Well, first of all, there are bacteria that are autotrophic. Trophic refers to food, and auto means self. So autotrophic bacteria can make their own food. Plants are autotrophic. Autotrophic bacteria, some of them are photosynthesizers, but in general, we find that they act as producers in food chains. Probably the best known group is a group called cyanobacteria. The cyanobacteria are sometimes called blue-green algae. Biologists are trying, but algae doesn't really have a very good scientific meaning. Algae usually means anything that's found in water. It's photosynthetic and it's small. And even those parts of the definition aren't really consistent. Now, notice cyanobacteria contain chlorophyll A. You might remember that chlorophyll A is found in plants along with chlorophyll B. And by studying genetic similarities between cyanobacteria and plants, there's pretty good evidence that cyanobacteria evolved into chloroplasts by a process called endosymbiosis. Symbiosis means living with another organism, so cyanobacteria and a plant cell. Endo means inside, because the chloroplasts are found inside the plant cell in the cytoplasm. And we'll talk more about endosymbiosis when we talk about the beginning of eukaryotic cells. We'll talk about those along with mitochondria, because mitochondria also seem to have evolved by endosymbiosis. Now, there are some filamentous cyanobacteria. And when we say filamentous bacteria, or filamentous anything, their body shape is a filament. So think of something like a worm. They have long, skinny bodies. And some of the filamentous cyanobacteria, for instance, there's one called Anabaena, have cells called heterocysts. And if you would look at a heterocyst under the microscope, a heterocyst has a very thick cell wall. Bacteria do have cell walls, remember, peptidoglycan. And heterocysts are very important because they fix atmospheric nitrogen. And they fix it, they convert it into ammonia, and then a few of them convert it into either nitrite or nitrate. And this is important because if we look at atmospheric nitrogen, what types of molecules are nitrogen found in living things? Does anybody know anything that contains that amine, or it's sometimes called amino group? Can anybody tell me a molecule that would have that? So there are a lot of different things that have the amine or the amino group. And so nitrogen is a crucial nutrient. If you look at most plant fertilizers, one of the main things they contain is nitrogen. Now keep in mind, if you would analyze the air around you, air is about 78% nitrogen, N2. But there's a problem. Plants can't use N2. They can't use atmospheric nitrogen. They can use ammonia. A few plants can use nitrite or nitrate. And so what things that fix atmospheric nitrogen do is they convert it into ammonia or nitrite or nitrate. To get it to nitrite or nitrate, usually it's a 2 or 3 step process. Usually the conversion is to ammonia. You might remember when we talked about photosynthesis, when we talked about the light independent reactions, we said that in the Calvin-Benson cycle, plant cells fix carbon dioxide. They take carbon dioxide out of the atmosphere, they put it into an organic molecule. So cyanobacteria are also found in connection with fungi. They make some things called lichens. And we'll talk about lichens a little bit more when we talk about fungi. I'm not sure, because a lichen is kind of a cooperative association between a cyanobacteria and a fungus. But for some reason, lichens are classified as fungi. They're not classified as bacteria. So I'm not sure why. Now there are a group of bacteria called the alpha proteobacteria, and this is this is actually, according to most people, the largest group of bacteria, according to most classification systems. It's kind of hard for us to say whether this is a kingdom or a phylum or exactly what it is. The classification system of bacteria, I think I mentioned before, biologists agree they're in the domain bacteria, the genus and species is pretty well agreed on. All the levels in between are really up for grabs. There isn't a lot of agreement about, where we should draw the lines for a family or for a order or for a phylum, or if any of those things are even relevant. Because remember what we said before, that system of classification into all those levels is really based on the idea that you have individual organisms that reproduce with each other. Well, think about what we just said. Bacteria don't reproduce with another bacteria. They simply split in half by binary fission. So it's not clear exactly where to draw these lines. Most people, when they think of bacteria, we think of parasites that cause disease. And those are certainly some of the ones that get the biggest headlines. But we have these alpha proteobacteria that one group of them probably evolved into mitochondria. We have the cyanobacteria that evolved into chloroplasts. So from a long-term biological perspective, they're important in this way. When we have bacteria that are parasites, sometimes they cause problems simply because they reproduce and their population gets so big, they interfere with what your cells are trying to do. Keep in mind, it's estimated that a typical human body has a little bit less than 40 trillion cells. And it's estimated that a typical human body has about 3 times that many. So about 120 trillion bacteria cells living in it and on it. Now, some bacteria cause problems because they make toxins. And toxins are literally poisons. And bacteria make 2 basic types of toxins. There are some toxins that they make that are endotoxins. Endotoxins are inside, they're inside the plasma membrane. And if we look at endotoxins, they're released when a bacteria cell dies and then the plasma membrane and the cell wall deteriorate. That releases this toxin. But bacteria that are most deadly tend to be the ones that produce exotoxins. Exo, think of exit. Exotoxins are released out of a living bacteria cell. So if we look at exotoxins, there are several bacteria that make exotoxins, and they open water channels in cells in your small and, even more importantly, in your large intestine. Now, one of the main things that your large intestine does with the food that you eat is it reabsorbs water. So if we open water channels, water actually flows into the large intestine. So what happens as a result of that? Diarrhea. And there are some bacteria that cause the loss of so much water, they can actually kill people by causing dehydration. Some of them can cause a person to lose 1 or 2 liters of fluid a day. And if you think about babies, for instance, a liter weighs about 2 pounds. If you think about a baby that might only weigh 15 pounds, if that baby loses 4 pounds of fluid, that baby is going to be on life support. For you, you could tolerate it. You'd have to drink a lot of water to replace it. But for babies, it's too big a percentage of their body weight. And if you're small and you lose that much water, that starts to change the concentration of solutes in the cytoplasm, and that can cause problems. So what types of species are beneficial? Well, we mentioned species that take nitrogen out of the air and convert it into ammonia. Bacteria act as decomposers in the food chain. They break down very complex organic molecules into simple molecules. And those simple molecules can be used by other living organisms, including plants. And a lot of bacteria live as symbionts. I mentioned that you have about 3 times as many bacteria cells in and on you as you do your cells in and on you. You have bacteria that live in your digestive system. Some of these bacteria help you digest your food and help you to avoid getting diarrhea. Some of these bacteria help your immune system fight off bacteria that might be in your food. Because no matter how well you cook your food, there's thousands of bacteria in every bite. And some of these bacteria actually make vitamins for you. There's one vitamin called vitamin K that in most adults is made by bacteria that live in our intestine. And we don't really have to get vitamin K in our diet. It's very rare for people to have a deficiency of vitamin K. And this is because the bacteria living in our intestine make so much, they release some of it as a waste product. So we scoop up that waste product, bring it into our body and use it. There are also a couple of amino acids that bacteria make that because they're made by bacteria, you don't really have to worry about eating in your food, or you don't have to worry about eating as much of them anyway. Remember, proteins are made from 20 different amino acids. So if you're going to build a protein, you have to have all those amino acids in your body. So some of those you get from bacteria. Now, besides what we see in nature, bacteria have a lot of uses in industry and technology. There's a lot of food that's produced using bacteria at one stage or another. If we look at yogurt, that's the main thing that we use to make yogurt. We take milk, you can take different portions of the milk depending on what type of yogurt you're making, and basically you give it to bacteria and you let the bacteria partially digest that. So when you eat yogurt, you're eating something that bacteria broke down some of the nutrients in the milk. Now, if you've ever looked at a yogurt container, some yogurt containers say contains a live culture. What does that mean, a live culture? That means the living bacteria are still in that yogurt. Now, keep in mind, the type of bacteria that are used to make yogurt are actually bacteria that are good for your digestive system. So you're not going to eat a container of yogurt and somehow get a rare disease. A lot of the antibiotics that we get in medicine, actually we get from bacteria. You might have heard of something called streptomycin, which is an antibiotic, and it's produced by a type of bacteria, Streptomyces. Now you might wonder, why would a bacteria make chemicals that kill other bacteria? Or sometimes they'll kill molds. But think about when a bacteria cell lands on a piece of fruit in your house, or when it lands on a piece of bread. What is the bacteria going to use that fruit or that bread for? A source of food. So producing antibiotics that kill other bacteria or that kill funguses around, that's a way of protecting its territory. If other bacteria can't tolerate this antibiotic because, the bacteria that makes streptomyces isn't affected by that makes streptomycin is the name of the antibiotic. That bacteria isn't affected by streptomycin, it makes it. So it's not harmed by it. But you can keep other bacteria or fungi away from your food. And I mentioned that, we've used plasmids, which are genetic pieces that come from bacteria in genetic research, but we've also used plasmids to try to put DNA into eukaryotic cells. And a lot of the successful genetic engineering that has been done so far has been done with bacteria cells. If you know anybody who's diabetic, almost all insulin that diabetics use now is produced from bacteria. These bacteria have the gene for human insulin put into their cells. Insulin is a protein. It's made of amino acids. So we put the instructions in bacteria cells. We let the bacteria cells reproduce, so we have a lot of them. And then we grow the bacteria cells in large vats. As far as the bacteria are concerned, they have no use for insulin. It regulates blood sugar. Bacteria have no blood, they have no blood sugar. So it's a waste product. So after they make insulin, they excrete it out of the cell as a waste product. We scoop it up, we purify it, and we have insulin for diabetics. And we have numerous other proteins that are made by the transfer of DNA into bacteria cells. Now, why bacteria cells instead of eukaryotic cells? Well, again, go back to something I referred to earlier. A typical eukaryotic cell, in the human cell, about 2% of the DNA is actually the genes, the instructions for making proteins. And that's if we count all 23,000 genes in a human cell. But a human cell only uses about 10,000 genes, so it only uses about half of that. So only 1% of the DNA in a human cell is actually going to be used in any particular cell. So this means if I transfer some DNA into your cell and I want the cell to use it to make a protein, I have to get it in the right spot. With bacteria, pretty much all the DNA is used to make proteins. So it's much simpler to just insert some DNA and the bacteria cell will probably make that protein. Now we're getting better at some tricks to make sure that eukaryotic cells make proteins, but it's still a little bit tougher. If we can get a prokaryotic cell to make the protein, it's usually easier, it's usually cheaper, and it's usually more efficient. We haven't had very luck if you have a disease where you don't make a protein. We haven't had very much luck changing the DNA in our human cells, whereas it's pretty easy to do with a bacteria. Are there any questions about the domain bacteria before we leave this page? No? Okay. We're going to switch to another page and talk about another group of organisms that used to be considered bacteria. They're not truly considered bacteria now. We classify them in a different group. They're classified as Archaea. And so let's look at the Archaea. And if we look at Archaea, Archaea are one of the three big domains of living organisms. Bacteria are one, eukarya are a second, and Archaea is the third. So let's look at Archaea. And Archaea, that name comes from the same root word as archaeology. And this is because when scientists first recognized that Archaea were probably a separate group, which really only happened about 40 years ago, about 1980, a little bit sooner than that. But when scientists first realized that Archaea were so different they should be in their own group, they were believed to be probably the first living organisms that evolved on the earth. So that's why the name Archaea, because Archaea means ancient. Since that time, we've discovered that that's probably not true, but the name stuck. Now, since they're in a separate domain, when they were classified with bacteria, they were actually called the Archaebacteria. Now, today we just call them Archaea, so we kind of dropped the bacteria off the name, off the end of the name. But the reason they were originally considered to be bacteria is they have prokaryotic cells. They don't have a nucleus, they don't have membrane-bound organelles like the endoplasmic reticulum, the Golgi apparatus, etc. So why would we think that the Archaea are maybe similar to the first living organisms that had evolved on the earth? Well, one of the reasons is because the first bacteria that were Archaea that were discovered and studied are what are sometimes called extremophiles. They live in very extreme environments, environments that are too hot, too acidic, sometimes too alkaline, too salty, anaerobic environments. Some of the types of environments that we think were probably common on the early Earth. What are some examples? Well, some Archaea are referred to as thermophiles. And most of the thermophile Archaea grow best in temperatures that are higher than 45 degrees Celsius. Keep in mind, 45 degrees Celsius is about 113 degrees Fahrenheit. Some of them can even survive in hot geysers, like you might see out at Yellowstone National Park or in Iceland. And in some of these areas, the actual temperature of the water in the geyser is higher than 100 degrees Celsius. Well, 100 degrees Celsius is the boiling point of water. So how could a living organism possibly survive in a temperature higher than the boiling point of water? Because we know that the main chemical that's found in cells is water. And so that would be the problem that a person would have. If we look at a person, keep in mind the boiling point of water is 100 degrees Celsius. For a person, typically for a person, water starts to feel hot somewhere around 33 or 34 degrees Celsius. If it gets up to about 120, that's unbearable for most people. So we've got some bacteria. I'm sorry, when I said 120, that was Fahrenheit. So that's only about 45 degrees, 48 degrees. That's unbearable to us. If you put your hand in water that hot, you wouldn't be able to leave your hand there very long. So how can thermophiles live in water that's literally boiling? Well, a couple of things. First of all, they make some proteins that work like antifreeze does in your car. If you've had a chemistry class, you might remember something called colligative properties. And with colligative properties, when we add other chemicals to water, it lowers the freezing point of the water and it raises the boiling point. So thermophiles make some chemicals that work like antifreeze. They raise the boiling point of the water in the cell. They also lower the freezing point. So they can live in a hot water where hot geyser where the water around them is boiling. How can they do this? Because they've got antifreeze. Notice there are also some Archaea that live in very cold environments in the oceans, in the Arctic and the Antarctic area. And in those super cold environments where the water is just barely above freezing, the Archaea seem to outnumber the regular bacteria. There are other Archaea called halophiles. So again, if you've had chemistry, you might remember the halogens. The halogens form chemicals that are often called salts. And halophiles can live in environments where the salt content is as high as 34%. Now keep in mind, that's much, much saltier than seawater. Seawater is typically less than 10%. So this is much, much saltier than seawater. And if you've ever been in the ocean and gotten a mouthful of water, you know that tastes very salty. Now, what's the problem that living organisms have in living in an environment where there's a very high salt content? Think back to Biology 105 when we talked about diffusion and osmosis. If you have an organism living in an environment with a very high salt content, water tends to leave the cell by osmosis. So the cell dehydrates. This is one of the reasons why a body of water called the Dead Sea is dead. The Dead Sea is so salty, there aren't very many organisms that can live in it. And when you get more salt in the water is also denser. So if you can't swim, you could take a trip to the Dead Sea sometime. And even if you can't swim, you can float on top of the water. The water is so dense, it buoys you up. Now, there are alkalophiles that live in alkaline or basic pHs. Some of them live in conditions where the pH is around 10, which is basically the pH of a lot of antacids that you might take if you have an upset stomach. There are acidophiles that can live in even more extreme conditions. Some of them can live in pHs as low as 0. And a pH of 0 is equivalent to, we keep going back to chemistry today, is equivalent to something in chemistry that we call 1 molar sulfuric acid. Now, how strong is 1 molar sulfuric acid? Well, if we have a little bowl of 1 molar sulfuric acid and we put your finger in it, your finger will literally dissolve. It will eat through your flesh. So we have Archaea that can live under these conditions. Today, as I say, we recognize that the first living organisms that evolved on the planet probably weren't Archaea, but they're sometimes studied to try to figure out what extraterrestrial life might look like, because they can live in such extreme environments. And also because some of them, if we look at the gases they produce, when we think of living organisms producing gas, we usually think of plants, and plants mostly release oxygen into the air, or we think of animals. Animals mostly release carbon dioxide. Some of these organisms make methane and other compounds. So this is one of the problems we have in general when we look for extraterrestrial life. What are we looking for? Well, something like us, of course, but that might not be what's out there. The other reason Archaea has been valuable recently is it's been used commercially. If you think about cells that can survive under high temperatures or very acidic conditions, this means they must have some special adaptations in an evolutionary sense. So what we've been able to do in some cases is we've been able to isolate enzymes from these Archaea that are very resistant to high temperatures, that don't break down easily. And high temperatures are often good in industrial processes because usually when you increase the temperature, chemical reactions happen more quickly. And in an industrial process, you generally don't want them to take a long time. So let's look at one of the characteristics of Archaea that's been the most studied that has to do with how they can tolerate these extreme conditions. And this has to do with their plasma membrane. So we're going to be comparing the plasma membrane of Archaea with the plasma membrane of eukaryotic cells and prokaryotic cells in the domain bacteria. Because Archaea do have prokaryotic cells, but it turns out their plasma membrane is not like the plasma membrane of bacteria cells. Even though they don't have a nucleus or any other membrane-bound organelles, the plasma membrane is very different. So let's think about some of the key differences, and then we'll see why they seem to be important. So if we look at, remember, glycerol has 3 carbons. If we look at glycerol, which is kind of the framework for building our phospholipids that we find in the plasma membrane, if we look at glycerol, it's a type of molecule called D-glycerol in eukaryotic cells and bacteria cells. In Archaea plasma membranes, in their cells, it's L-glycerol. Now, what's the difference between L-glycerol and D-glycerol? Well, L and D glycerol are two examples of something that we talked about back in Biology 105 when we were talking about why carbon is important for living things. One of the things we said was that carbon can form chiral molecules, molecules that are basically mirror images of one another. L-glycerol and D-glycerol are mirror images of one another. So that's one of the reasons that's important. Now, there's another reason that the plasma membrane is different. When we look at eukaryotic cells and bacteria cells, and I'm going to make this bigger again so that we can see what I'm talking about in our little diagrams of the molecules. If we look at eukaryotic cells and bacteria cells, the connection between these unbranched tails. And remember that these come from fatty acids when we're looking at eukaryotic cells and bacteria cells. And the glycerol is the yellow here. Notice that the connection between the fatty acids and the glycerol is through something called an ester linkage. And this is a carbon that has a double bond to an oxygen, and then that oxygen is bonded to another carbon. Remember, oxygen needs two bonds. And that carbon also has a double bond to O attached. And then over here, it's bonded to the next carbon in the chain. Remember our way of representing organic molecules, that at every vertex, there's a carbon atom with as many hydrogens as it needs to get its 4 bonds. So when we look here, we have an ester linkage and we have fatty acids that are unbranched. Remember, with carbon compounds, we talked about carbon compounds could form rings, they could form straight chains, or they could form branched chains. So an unbranched tail is a straight chain. Now, if we look at Archaea, they have L-glycerol, and then they have something called an ether linkage. And the ether linkage is a carbon connected to an oxygen, connected to another carbon. But notice there's no double bonded oxygen on these carbons in the purple. So there's another difference. Okay, so the type of glycerol, the type of connection. Now the third difference is these tails, and notice these would be the non-polar parts of our plasma membrane. These tails are from straight chain fatty acids, if we're talking about a eukaryotic cell or a bacteria cell. If we're talking about Archaea, they're actually from little 5-carbon groups called isoprenoid groups. And these isoprenoid groups are branched. So notice one of the things this means is that we've got some little carbon branches sticking out that sometimes get tangled up in a neighboring isoprenoid group. So we'll come back to that in a moment. So 4 differences so far: different types of glycerol, different types of linkages, unbranched versus branched, and fatty acids versus isoprenoid groups. And there's still one more difference. Here's an isoprenoid group, by the way. If we look at, and this is kind of a repetition of the same information, but if we look at a plasma membrane from a bacteria cell or a eukaryotic cell, remember that we said that the plasma membrane was a bilayer of phospholipids. And we had one layer facing the inside of the cell, one layer facing the outside. And then we have 2 layers of molecules. And these 2 layers of molecules can slide across each other. If we look at Archaea, there's a difference. For the archaea, now this is showing it as every molecule in the plasma membrane, but it's not necessarily every molecule. It's usually just some of them. Notice here, we have one molecule that spans the whole width of the membrane from the outside to the inside. And this happens because sometimes the isoprenoid groups are connected to a phosphate on this side, and they're connected to a phosphate on this side also. So this means that we have a molecule that's kind of a mirror image of itself if we split it in half, excuse me. But this means some molecules go all the way through the membrane instead of all the molecules going just halfway through the membrane. Okay, so why do all these differences matter? The type of glycerol, the type of linkage, the type of molecule here, whether it's branched or unbranched, whether it goes all the way through to the other side or only goes halfway through. Well, the reason they matter, and if you think about it, this makes sense, but when we started studying the plasma membrane in Archaea, the plasma membrane in Archaea is much more stable. It's a lot sturdier than this phospholipid bilayer. And it's all these different factors. It's the fact that we have a different type of glycerol. We have a different type of linkage. We have branched isoprenoid groups. We have some molecules going all the way through. This means that the plasma membrane is much more stable. Now, why does that matter? Well, because these organisms live in areas where temperatures are very high or acidities are extreme, or alkalinities, depending on the environment. So how can they tolerate these kind of extreme chemical conditions? Well, easy. The way they can tolerate these extreme chemical conditions is by having a plasma membrane that's much more resistant to chemicals, to temperatures, and things like that would damage the plasma membrane in your cell or a typical bacteria cell living in your body or on your body. So the plasma membrane is something that's very different from what we see in bacteria or eukarya. Now, the reason that Archaea were first classified with bacteria is because the most obvious thing we can notice when we first look at them is they're prokaryotic cells. They don't have a nucleus. They don't have membrane-bound organelles. But it turns out Archaea are not that closely similar to bacteria. Remember, in bacteria, the cell wall is made of some chemicals called peptidoglycans. That's not what the Archaean cell wall is made of. I can't tell you one chemical because even though almost all bacteria cell walls are peptidoglycans of one type or another, there are several different types of Archaean cell walls. And when we start looking at the biochemistry, if we look at the replication of the DNA, if we look at transcription of RNA, if we look at protein translation, so some of the processes that are crucial for living things, if we look at those, the metabolic pathways to do those things are closer to what we see in eukaryotic cells than what we see in prokaryotic cells. And some things are kind of half and half. If you would look at a ribosome in an Archaea cell, the ribosome is actually the same thing, same size, I should say, as in a bacteria cell. But even though it's the same size, the ribosome in an Archaea cell doesn't work like the ribosome in a bacteria cell. It works like the ribosome in a eukaryotic cell. So because of this, when we look at all the similarities and we classify the Archaea are not classified with bacteria anymore. In fact, if we look at how bacteria and archaea and eukarya, the 3 domains, are classified, now think about what we talked about the other day when we were analyzing cladograms. The bacteria split off first. So remember, the group that splits off first is least closely related to the other groups. The groups that split off more recently are more closely related. So the archaea and the eukarya split off a little bit after the bacteria have split off. So these two groups are more closely related. And this is one of the things we always have to be cautious about in studying biology. We tend to look at things, for obvious reason, from a human perspective. That's not necessarily the best way to analyze organisms that are very different, is to think about things from a human perspective. In this case, for many years, it led to a kind of a tree of life that had true branches. You know, if we wanted to go back to what we used to think, we could just move this purple branch over and have it coming out of the blue branch somewhere, because the archaea were considered to be one particular type of bacteria. So we have to keep that in mind. So today, there are a lot of archaea that haven't been very well studied, because if you think about what I've said, a lot of archaea live in very extreme conditions. Well, in laboratory conditions, it's very hard to raise these organisms that live in water that's hotter than the boiling point of water, or that live in, you know, very strongly acidic conditions or very strongly alkaline conditions. So a lot of archaea haven't been studied very well. And as we learn more and more about archaea, we're finding them in new places where we didn't find them before. And even though a lot of archaea do live in extreme conditions, there are some archaea that live in the human body. You know, if you've ever had carbohydrates like beans, and then you've later produced some gas that reflects that you couldn't digest all those carbohydrates, that gas that you release after you eat something like beans, or some people do it when they eat broccoli, that's produced by bacteria in your digestive system that can digest those foods. So there is one group of archaea called methanogens that make methane, and methane is an ingredient in natural gas. So you can find out if you have methanogens in your digestive system. Eat some beans, wait till the appropriate time when you're about to release some gas, and light a match back by your rear end. And if it produces a blue flame, you're making methane. So notice some animals, like cows, actually produce large amounts of methane, because cows have way more bacteria living in their digestive system than you do. Does anybody know why this is a problem with cows having so many bacteria that produce methane?
Is it bad for the environment?
Yes, it is. You know, now I'm going to ask my follow-up question. Do you know why it's bad for the environment?
Probably something with the ozone layer. I don't know.
Close. Not the ozone layer, but global warming. Turns out that methane, you know, the main gas that we're worried about with global warming is carbon dioxide, because carbon dioxide absorbs more solar energy than nitrogen and oxygen, the two most common gases in the air around us. Turns out that methane absorbs about 10 times more solar energy than carbon dioxide does. And so a lot of people think that one way that we could potentially stop global warming is by having people eat less beef. because if people were eating less beef, we wouldn't be raising as many cows. We wouldn't be releasing as much methane into the air. Okay, are there any questions about Archaea? Okay, if not, we're going to leave this topic. And we have one more thing we're going to talk about a little bit before I let you go. So actually we have two more topics. One is not going to take very long. Okay, let's go to, okay, I think you should see a long list of bacteria and Archaea. This is some of the genera. Remember, genera is plural of genus. This is some of the genera. There are a lot more than this. We'll talk later, but this is posted right now. And one of the things you may want to start looking at, I'll give you some more specific instructions tomorrow, but start thinking about if you have to write an essay question, which one of these genera you would like to write an essay question about. Everybody in the class doesn't have to pick the same one. But when we get to our test on, or I should say our questions on bacteria and Archaea, and we still have one more group to talk about, bacteria and Archaea and viruses. When we get to that, then you will have to answer an essay question about one of these groups of bacteria. Now you can write that essay question ahead of time and you can just cut and paste it or send it to me as a separate document. But I wanted to post this today just so you could start looking. So this is a variety of different genera. Some of them are things that cause diseases. Some of them are things that live cooperatively. Some of them are Archaea that live in strange environments. So think about one of these that you might find interesting enough to do a little bit of research on. Because this is kind of a combination. It's on the bacteria, virus, Archaea questions. This is kind of the essay question, and it's also the first paper that the syllabus mentions. So I'll give you a lot more detail on what you're going to have to do tomorrow, but start thinking about, something that you might want to write about. It's not going to be a very long paper. It's going to be a page, so not real long. Any questions? Okay, I want to look at one more thing before we go, which is I want to look at some of the Hardy-Weinberg problems, because you will have to work on one on the exam. So let's look at a problem, and let's look at probably the most common example where people miss some points. So think about number 6. So notice it's asking us some questions about the phenotype and then about big B. So let's go down to number 7. Let's work a different one. Okay. So here, big S is short hair, little s is long hair. So what's the frequency of the big S allele? So the first thing you have to think about when you see this is big S is equal to 1 minus little s. So even though it asks you big S first, you can't figure that out. You're going to have to go down here and you're going to have to figure out little s. So what do we know about little s? Well, what we know about little s is we're generally going to start with the phenotypes. So little s squared is equal to 289 divided by 10,000. But notice this is asking us little s, not little s squared. Does anybody remember how we're going to figure out little s from this?
Take the square root.
Okay, we're going to take the square root of both sides. And I think we're going to get 17 over 100 if I did my math right. So notice in this case, you have to do B before you can answer A. What a lot of people did, you know, if you look at the frequency of the black phenotype and then the frequency of the big B allele, how did they find big B? They took the square root of their answer for A. Can't do that. If you take the square root of that, you're not going to get big B, because remember what we said, the problem is if you're looking at the frequency of the black phenotype, that category includes two different types of genotypes. It includes some guinea pigs that are big B, so they have black hair. It includes some guinea pigs that are big B, little b, so they still have black hair because big B is dominant. But if you take the square root of this number, that means you're assuming that all the guinea pigs that have black hair are big B, and that's not true. So you won't get the correct answer. You'll almost always get an overestimate, but not always mathematically. The other question that I saw people miss was question like E, how many cats out of 18,000 are heterozygous for hair length? So remember, our frequency for hair length is 2 times big S times little s, but then since it's how many, you have to multiply by population size. So that's going to be 2 times, I guess I should keep them in the same order, 2 times 0.83, which is big S, times 0.17, and it's our population size is 18,000. And then remember, when we multiply all this out, you have to round this to a whole number. You can't have 0.8 cats at the end. So round up or round down, depending on what number you get, but don't leave it as a decimal. And similarly here, how many copies of the big S allele are there? Oops, how about times population size? Because remember, we said there are 2 copies of the allele in each individual. So you have to take the population size into account and the fact that everybody has 2 of these. So on the set of questions that I should be able to post later today, because it's somebody told me the secret of giving you an assignment worth more than 100 points. So should be up in another hour or two. So on the Hardy-Weinberg problems, it will say to show all your work. So show me the formula that you're using. Show me the numbers that you substituted into your formula. And then, of course, give me the answer, multiply it out. And all the questions on the Hardy-Weinberg and also on the natural selection will tell you to, round off to the third decimal or the second decimal or something like that. So that then we'll all hopefully get the same answers that way. So any questions about how you would work some of these?
Rounding to the whole number, do we round up as well?
So kind of the same rule that we usually use in math. If it's 0.5 or above, round up. If it's less than 0.5, round down.
Okay, sounds good.
Just make sure that you don't just truncate numbers and say 14.9. Well, that's 14. Any other questions? Okay. I'm going to stop sharing now, and I'll go ahead and I'll let you go, and I will see you again tomorrow. And like I say, the questions will be posted in a little while. The video will also be posted in a little while.
Professor, I have a more general question.
Sure.
So I came across a question on a test on another class. It was a true or false question.
Mm-hmm.
And it was about prokaryotic cells.
Mm-hmm.
And it was true or false, prokaryotic cells do not have organelles. And I hit false because I didn't see membrane bound in there.
Yeah, I would agree with false. I would say they don't have membrane bound organelles. That would be true, but really the ribosome is a type of organelle. The chromosome is a type of organelle. The flagellum, the pilus. Some people even consider the cytoplasm and the plasma membrane organelles.
But that was it was marked incorrect, and it was I was told that ribosomes aren't organelles, and that was my example that I gave. And I was just doing some extracurricular research, and I didn't really find a clear answer on that.
Yeah, I would agree with your answer, because I think most people do consider ribosomes to be organelles. Like I say, a lot of people also consider the cell wall, the plasma membrane, flagella.
Did he say membrane bound organelle or just organelles?
If it just said organelles, I would say it was false. If it said membrane bound, then I would say it would be true.
Okay, so he said prokaryotes, they have membrane. I didn't get the question well.
I think he said the question was just, true or false, prokaryotic cells do not have organelles. So I would say that's false, because they do have ribosomes, and that's an organelle. And as I say, some people consider the plasma membrane and the cell wall and the cytoplasm and the chromosome, some people consider those to be organelles also. And the flagellum and the pilus. And
I'm thinking is it true? Because it says prokaryotes do have organelles, right? Do not have organelles, right? True
I think that was the question. So I would say if it said they do not have membrane bound organelles, I would say that's true, but I would say it's false that they don't have because if the question is they don't have organelles, I think that's false, because they do have ribosomes, and that's organelles.
Yeah, do not have organelles.
I think you would have to say I think for that to be true, you'd have to say they don't have membrane bound organelles. That's how I would interpret that anyway.
Yeah, that's how I did as well. Thanks.
Yeah, I think I would agree with your interpretation.
Questions are confusing. You have to analyze before you answer those questions.
Well, you have to keep in mind on questions. A lot of times it's one word or two words make a big difference in how you interpret it.
Right.
It would be like with living things if you would say, all cells have a nucleus. Well, that's not true because prokaryotic cells don't have a nucleus.
Right.
But if you said some cells don't have a nucleus, then that would be true. And I think that's one of the things that takes some adjustment in science compared to some other academic fields. And a lot of academic fields, I think they're not maybe as detail oriented in terms of, it doesn't have that big an impact on your final answer.
Biology especially, biology is always like that. You must understand what you want to do. If you don't understand it, it's all probability of you missing the question. You have a high chance of missing the question if you don't understand what you're doing.
Yeah, and I think that's why it's always good to first kind of analyze the question. I mean,
Right.
It's a lot harder when we have our tests in class because then there's a shorter time limit, but I think it's not a bad idea when you get the question sets to kind of go through and read all the questions and maybe put them aside for, a day or half of a day and so that
Come
you
back
have
to
some
it and
time
read
for
it
it to
again.
sink in.
Right.
Because sometimes if you answer things, if you do it as soon as you look at it, you aren't reading carefully, you jump to some conclusion or something. Because I know sometimes I've had people tell me when we talk about tests, well, the reason I answered this was because if this happens, then that would be correct.
Yeah,
And I'll say, but does the question say that happened?
Yeah, we're looking at just one point of view, just looking at one specific part. And
yeah, it's like, kind of stick to what the question tells you and asks you.
Right.
But that's a good thing if you're a teacher. You get to hear all sorts of unusual answers over the years. I had a student a few years ago who came up to the desk. This was before COVID. We were still on campus. They came up to the desk during a test and they said, "What if you like answer B for question number 2?" And I was like, "Well, then mark answer B." And they said, "Well, but here's the problem. I like answer B on question 3 for number 2." Why are you looking at the answers for number 3 to see if they're good answers for number 2? I don't get this. So sometimes people surprise you.
Right.
I had a student on a multiple choice test one time who asked me, "Do you want us to answer A or B or C or D?"
That is why you're giving the question.
It's like, okay, you're 20 years old and you've never seen a multiple choice questions before? Yes, answer A or B or C or D.
The question sometimes can be confusing. You would be like, what? Oh.
Well, if you don't understand a question, I can sometimes explain to you what I mean, but I try to avoid giving away the answer if I do that.
Right. So those questions, I see them as standard questions though. Like you want to go back and look at it again, another point of view, This is get to one part.
And for sections like we're on now, if you don't remember, I mean, I always tell you, go back and listen to the recording again, or go back and look at the notes again, or look at the book. But the other thing you could do on a section like this is, we keep mentioning eukaryotic cells, but I haven't really talked about them a lot. But that's because I kind of think that you know about eukaryotic cells from 105. But if you don't remember 105, it would be good to go back and look at, I think that was chapter 4 in 105 about cells. It would be good to go back and refer to that too. Because I try to do a little bit of review, but if we do too much review, we'll never get to the new stuff is the problem.
Right. Biology is too big, too wild.
Yeah, it's hard. And unlike some fields, basic biology is always growing every year. I think, basic physics and chemistry are still pretty similar to when I took them as a student, but, we have a lot more things about DNA that we know now every year.
Things change like almost every time. Yeah. All right, Mr. Griffith.
Bye-bye. We'll see you tomorrow.
Yeah. Bye.
Any other questions or comments?
All right, take care.
You too, Nick. I'll see you tomorrow.
Bye.